All right, let's dive into this explainer. Today, we're putting on our detective hats and acting as forensic botanists to solve a massive, real-world ecological mystery. We're going to be looking at the completely invisible, microscopic architecture that literally holds our coastlines together. We're talking about coastal macro-vegetation, mangroves and seagrasses. These plants act as the biological armor of our coastal ecosystems. They stabilize the shores, they filter out sediment, and they provide absolutely crucial nursery grounds for fisheries. But to truly understand how they manage all that, we have to look incredibly closely.
Now picture this for a second. We are investigating a catastrophe of epic proportions. 95% of 15,000 newly planted red mangrove seedlings, specifically the species Rhizophora mucronata, died within just 2 months. And this wasn't some tiny backyard experiment either. This was a high-budget municipal coastal rehabilitation project out in low-lying intertidal flats. Obviously, an environmental audit was called in, and what they found was just shocking. The roots of every single one of these plants had completely rotted away.
So you gotta ask yourself the big question here. Why did 15,000 aquatic plants drown? I mean, it sounds like a total oxymoron, right? Well, the planting team basically assumed that because mangroves are aquatic plants, they could easily handle indefinite submergence. So they just planted the seedlings in deep, artificial mud basins that stayed completely flooded, even during low tide. They completely ignored the microscopic anatomy of survival. They forgot a really fundamental rule of botany, which is that root tissues are metabolically active. They absolutely, 100% require exposure to atmospheric oxygen to perform cellular respiration.
Moving into Section 1, Anatomy of Breathing Roots. To actually solve this mystery, let's head straight to the autopsy table and figure out what went wrong. We need to understand the microscopic structures that keep a mangrove alive in oxygen-starved, waterlogged mud. To survive in those brutal anaerobic sediments, mangroves rely on highly specialized aerial roots, like the prop roots you see on the red mangrove. And right on the outer protective layer of these roots, we find these structures called lenticels. These are crucial. They are microscopic pores lined with hydrophobic waxes. That wax is the key here. It actively repels liquid seawater. But the moment it's exposed to the atmosphere during low tide, it allows oxygen gas to pass right on through. Okay, so catching air at the surface is great, but how does that oxygen get down to the roots buried deep in the mud? Well, enter aerenchyma tissue. If you take a cross-section of a root, just beneath the outer layer, you'll see the bulk of it is made of these thin-walled, spongy cells around large circular cavities. These channels serve as internal ventilation tunnels. Think of it like natural plumbing. It's literally a low-resistance airway that transports the oxygen gathered by the lenticels all the way down to the submerged root tips. It's a wildly impressive piece of biological engineering. So here is where the blunder actually happened. By planting those seedlings way too deep in artificial basins, the team doomed the project right from day one. Let's trace the domino effect here. Because the lenticels were permanently underwater, they couldn't capture any air. Water and fine mud silt breached the system, basically infiltrating and physically clogging up those delicate aerenchyma channels we just talked about. Cut off from oxygen, the root cells couldn't generate energy normally. So in a desperate bid to just stay alive, they switched to anaerobic fermentation. And that led to a massive, lethal accumulation of toxic ethanol inside the plant tissues, which caused systemic root rot and ultimately killed the plants. They literally suffocated them. Which brings us to Section 2, seagrass, the submerged survivors. Now, what about marine plants that thrive completely underwater without ever touching the air? Let's take a look at seagrass species, like Angelus ovalis. Seagrasses basically throw the standard botany rulebook right out the window. When you compare them directly to terrestrial plants and mangroves, it's pretty wild. Normal plants keep their light-harvesting chloroplasts safely tucked inside the leaf. But seagrasses, they pack them right on the very outside, the chloroplastic epidermis, to catch every single possible ray of light in deep, cloudy water. Also, they don't have any functional stomata or lenticels. Instead, they use an extremely thin, non-waxy cuticle to just absorb dissolved carbon dioxide straight from the seawater. And because they absorb water directly through their leaves, they don't need heavy vascular plumbing to pull it up from the roots, so their structural xylem is highly reduced. But wait, if their structural xylem is reduced, how do they stay upright against ocean currents? Well, they use aerenchyma lacunae. Instead of dense, stiff wood, seagrasses use these oblong, internal gas-filled balloons. These chambers store oxygen produced during photosynthesis, but more importantly, they provide vital hydrostatic buoyancy. It's exactly this internal pressure that keeps their leaves floating perfectly upright in the water column, constantly reaching toward the sunlight. All right, section 3, slicing and staining tissues. To actually see all these crazy microscopic adaptations, like the clogged aerenchyma in our failed mangroves, or those lacunae balloons in seagrasses, fisheries biologists have to do some incredibly precise lab work. First things first, before we even touch a blade, we have to lock those delicate cell structures in place so they don't shrink. We use a botanical fixative called formalin acetoalcohol, or FAA. Once fixed, we follow a super strict 4-step protocol. First, slicing the tissue with the microtome. Second, applying our dual stains. Third, mounting the sample in a glycerin medium. And finally, getting that pristine slice under the microscope for analysis. Now, step 1 sounds easy, right? Absolutely not. The rule of translucency means our slices have to be only 1 or 2 cell layers thick. Any thicker and overlapping cells just completely block the microscope's light. And slicing mangrove roots can be an absolute nightmare. Think about it. These roots have to anchor whole trees in shifting, high-wave ocean mud. To pull that off, they're packed with these thick, highly lignified sclerenchyma cells, basically heavy-duty structural fibers. Amazingly, these fibers are so incredibly tough, they can actually deflect and tear right against a soft steel microtome blade, just folding and ripping your pristine slice to shreds. But assuming you get a good slice, it's time for the dual stains. And this chemistry is basically magic. We use safranin O, a cationic dye that aggressively hunts down lignin. It binds to the tough secondary cell walls, like those incredibly hard sclerenchyma fibers and xylem vessels, staining the plant's structural armor a vibrant, deep red. Then we hit it with fast green FCF, which is an acidic counterstain. That one binds to the unlignified primary cellulose walls, turning all the soft, metabolic, and photosynthetic parts bright green. Instantly, under the microscope, you get this amazing color-coded map separating the red structural support from the green metabolic engines. The absolute last hurdle is mounting the slide, and this is where the 45-degree angle rule comes in. When you apply your glycerin medium, you have to lower the glass cover slip super slowly, exactly at a 45-degree angle, to gently push the air out to the sides. If you get sloppy and just drop the cover slip flat, you'll trap air bubbles. Under high magnification, those bubbles form dark spherical refractive rings. To an untrained eye, they look exactly like actual plant vacuoles or bizarre cell walls. You'll literally ruin your own forensic analysis because of a tiny air bubble. Finally, section 4, saving our coastal ecosystems. So we've explored all this microscopic cellular architecture, but why does slicing up plant tissues actually matter to a regional fisheries officer working out in the real world?
Well, it matters because it literally dictates how we manage and save these coastlines. Once you understand these tiny structures, you can spot root suffocation from farm silt runoff way before it becomes a disaster. An officer knows that fine silt will clog up those lenticles, so they can design better green belts. For instance, planting a protective buffer of salt-tolerant grasses, like Scyphophora, upstream to catch the sediment first. We can even use these stains to biomonitor heavy metal pollution, seeing exactly where toxins are building up inside a seagrass's vascular bundles. We can even observe the active transport pumps in salt-excreting glands that literally push toxic sodium out of the plant so it doesn't die in seawater. And that really leaves us with one massive, lingering question. If just blocking a single microscopic pore, a lenticel no wider than a strand of hair, can suffocate 15,000 trees and completely derail a high-budget afforestation project, how well do we really understand the hidden mechanics protecting our coastlines? I hope this explainer leaves you marveling at the incredible microscopic engineering of these plants. It is a powerful reminder of just how fragile and deeply interconnected this biological armor truly is, and why digging into the tiny details is really the only way we can protect the massive ecosystems we all rely on.